How's everybody feeling though? Okay. It's actually, I would tell you, this is a good test to grab grades because there are 5 lectures and you have more questions. Believe me, get good grades in exam 1. Exam 2 has a lot of lectures. This is the best time. Build the buffer in this exam. Tired, I understand. I'm tired too. I'm good, Amanda. Hope you're well. Let me know when it's 10. I will do, Amanda, those who missed the review after inheritance, okay? Keep staying. Anyone wants to stay, stay. Oh, I think uh sitting on the computer back hurts, believe me, all day. Even neck. You people should, get up and do some neck exercise and, nowadays, they recommend to stand up and do work on the laptop or computer for a few minutes. That helps straight back, right? I think it's almost 10, everyone, or no? We have a minute. Let's see who's ready. Oh, yes, neck, absolutely, Amanda. I think stand sometimes and do your work back straight. It is 10, everyone. Don't get stressed, please. This is the time to reflect. You must have watched the lectures. Let's do the focus. I'm doing inheritance first. My aim here is to, you can stop and ask me questions. So I'm doing it a different way so that at least we will done with the inheritance. Then I will do composition and replication repair. First of all, can you see my slides? Inheritance pattern recap, everyone? And can you hear me well? Okay, everybody. I'm seeing chat. Yes, everybody's yes. So let's first do this thing. First, you think you should do is identify an inheritance pattern. I told you first rule out mitochondrial. Now, this looks mitochondrial to me, E. Mother has the disorder, everyone has the disorder. Just give me a second. I have to keep scrolling this chats and everything. So I hear the questions. So what is E? Mitochondrial. First and foremost step is to identify the pedigree inheritance pattern. If the pedigree is not given, examples. As I told you, just to clarify, you don't need to know clinical presentation of inheritance pattern, please, because many have not been taught to you. But yes, know the disease and the gene. But this is only I'm saying for inheritance pattern. For other disorders, replication, repair, please no clinical presentation. First, you rule out, mitochondrial. Mom has the disorder, all children have the disorder. Dad has the disorder, none of the children have the disorder. E is mitochondrial. Now, X-linked is also confused with mitochondrial because maternal. So let's see how we can identify X-linked, which is A. In X-linked, we have taught X-linked recessive as well as dominant. I will do both, though I told you first do recessive in detail. 4 pedigrees I've told you to do in details. Autosomal recessive, its characteristics, example, autosomal dominant, X-linked recessive, and mitochondrial. But I'll explain you all. We have through this question. Excellent question. If you want to practice just before your test would be great. E is mitochondrial. Now let's go to X-linked recessive first, because remember X-linked recessive will more commonly be asked. Now X is also having maternal inheritance. First of all, X-linked recessive is recessive. So you see skipping of generation. But what tells you that this is surely X-linked recessive? Which question is this? This is my review slides. I will post that. This is from your lecture, please. This question is from your lecture. Okay. Don't worry. I'm going to post the review slide. Pay attention on the focus. Please focus now. Zen, right? It is from your lectures. So when you say A, There is skipping of generation. And do you see male more affected than female? Yes. Then this is A is X-linked recessive. Absolutely. A is X-linked recessive. Now let's go to the dominant also because we want to differentiate X-linked recessive from dominant. Why C is dominant? Because females or daughters are more affected. Females more affected and there is no skipping of generation. C fits X-linked dominant. So please, when a pedigree is given, don't waste your time in drawing Punnett square. You will lose time for other questions. All you need to see overall, rule out mitochondrial as I did for E. Maternal also is X-linked, so I distinguished A and C. How A and C are different. A, skipping of generation, so it's recessive and male more than female. I will get to other characteristic too, but you don't need Punnett square here, please. Why C is X-linked dominant? Female clearly more affected than male, right? Now what is left is B and D. So B and D, where do you see double line? That has to be recessive. Are male, female equally affected? So this will be autosomal recessive. D would be autosomal dominant. Why? Male and female almost equally affected, no skipping of generation. Now let's do exceptions, characteristic. Are you clear with that? Any question? I won't go too fast here because please see this is question and answers for you too. So first do that for me. Let's go further. If I see any question, I will answer that. You can unmute or also ask. Now do the characteristic. First identify, then do characteristic, then no example, then do calculations. So you know, yes, anyone has a question? It's best you unmute. To what A was on that slide. So for A was X-linked recessive. Am I right? Let's go to X-linked recessive. For A, I said X-linked recessive. Why? First of all, it is recessive because of skipping of generation and male more affected. So that is why it's X-linked recessive. Now, why it is not Y-linked, I'll show you. Keep it that, keep in your Zen. I'll also show you Y-linked. But this is clearly X-linked recessive. Did I answer you? Right? Let's go further. So when you are saying, okay, great, you're happy, I'm happy too. So when you say recessive disorder, you need only both mutant allele. So what are heterozygote in recessive known as carrier? Please, please, and please, in X-linked recessive, male are not carrier. In autosomal dominant, you do not talk about carrier because autosomal dominant heterozygote express the phenotype. So nowhere you don't talk about carriers. You talk about carriers in autosomal recessive? Absolutely. You talk about carriers in X-linked recessive? Absolutely. But only females are carrier. Let's do dominant, then do characteristic difference between each one of them. First of all, how do you say it's dominant? Male and female equally affected. Male and female equal chance to pass mutant allele. There is male to male transmission. If you see male to male transmission, you cannot pick X-linked, please. Right? Secondly, what is the mutant allele? Mutant allele in autosomal dominant is capital A. You assume that when it's a mating between, for example, here, this is a mating between an autosomal dominant affected parent and a normal parent. You have to assume, unless it's specified to you, that the genotype of the affected parent in autosomal dominant is capital A and little a, then draw your Punnett square. Does that make sense? Right? What else is for autosomal dominant? Autosomal dominant, this is clearly an autosomal dominant pedigree, but can you see one or two skipping of generation? Why not? So skipping of generation in autosomal dominant is because of three reasons: delayed onset, incomplete penetrance, and spontaneous mutation. Do you need to understand these terms? Absolutely, because this is a generic test. But try and understand that I'm addressing incomplete penetrance, delayed onset, and spontaneous mutation can be everywhere, but incomplete penetrance and delayed onset mainly for autosomal dominant disorder. right? So don't mix and match these terms. For example, incomplete penetrance. Don't talk in recessive disorder, please. So mates with normal parent, we have to assume normal strand, non-phenotype, but also. So when mating between what normal parent, Diego? So when is the mating between? Oh, so if in autosomal dominant disorder, the other parent is normal, we have to assume it's capital A homozygous. Yes. Right?
No, more like in a homozygous recessive. Sorry, autosomal recessive. And
Let's stick to
do
dominant first. Let's stick to dominant first,
okay.
right? I'm discussing dominant here. We'll get to recessive. One thing also will help you, please, that when you are doing inheritance pattern, take one inheritance pattern, complete it, then start another. So you're very clear with that. Right now, I'm doing autosomal dominant. I'll keep your question for recessive, Diego, bring it up. But for autosomal dominant, when you say that there's a mating between affected parent and unaffected parent, that unaffected parent genotype should be little a, little a. Affected parent genotype should be capital A, little a. Yes, somebody has written there because in autosomal dominant homozygous, which is capital A is lethal. But please, please, and please, when you're asked to draw a Punnett square between an affected parent, which is capital A, little a, and an unaffected parent, which is little a, in Punnett square, keep the children or offspring alive. They are not lethal. You have to count them all, right? Does that make sense? Only assuming what is the genotype of the affected parent? It should be capital A, little a. Example only with disease and gene. Clinical presentation for inheritance, you don't need to know. Please be familiar with these terms in your yes, somebody has a question. Please be familiar with these terms. I'm falling my place. If the in dominant we can mate with, so that is also a very good question, which I think I should go back also to tell you that very good question. Somebody's asking what could be the dominant possibilities. So yes, in autosomal dominant, common mating could be between affected parent and the normal parent. But look at this. There could be uncommon mating too, though autosomal dominant disease are not very common. So you can't see two parent affected. But yes, you should also draw a Punnett square and see what if the both parents are affected, which is not in this case. But no, you are not assuming that they are capital A, capital A. Is that if it's an affected autosomal dominant adult phenotype, you are not assuming it's capital A, capital A. It has to be capital A, little a. Is this your question? No, no, no mating with capital A, please. No. Only when you draw the Punnett square, count all the babies alive or offsprings alive, please. Right? So look at the common and the uncommon matings too. Be familiar and draw them so you don't need to waste too much time when you are asked. Practice makes man perfect, right? I'm giving you focus. More you practice, better your grades. But please stick to the focus. Yes. You want to look at delayed onset income. Okay. Okay. That's good, Andrew. Keep going. But yes, know that you talk about incomplete. That's great. Whatever helps you give me good grades back, right? So yes, in autosomal dominant, you talk about incomplete penetrance, not in recessive. And let's understand this concept with Huntington's disease. Huntington's disease is autosomal dominant. Look at this. There is still skipping of generation. That is possible in dominant disorder. Still, every generation should show a phenotype. There would be one or two skip. That's okay. That's acceptable. This doesn't become autosomal recessive, right? Secondly, please know a good example in autosomal dominant that there's a genotype but may or may not present with a phenotype. That's known as reduced penetrance or incomplete penetrance. Let's understand reduced penetrance and delayed onset from this question. This is the, please don't mix these two. This is the comparison of the pedigree above and the lane below. For example, lane 5 in the gel correspond to the individual 3-2. So you have to be mindful. So let's see who is showing incomplete penetrance and why, and who's showing delayed onset, and who's showing develop the disease later. Whenever there's a genotype, there should be a phenotype. This is autosomal dominant, so oh, there are two alleles, but only one allele will show expansion. Let's look at lane 5. There is a genotype, expansion, and phenotype. This is not a problem. Onset, good, 43. Look at lane 6, individual 2, 2. That corresponds to individual 2, 2. There's a genotype. You only need one allele to show the expansion, which should be 40 and above, but there's no phenotype, and which is not common to Huntington's disease. This is known as incomplete penetrance. Let's go to lane 8. You see the expansion, you see the phenotype, which is okay, but the age is early. This is anticipation. What is anticipation? With every successive generation, expansion increases. Yes, it's anticipation. And the disease is earlier onset. Let's go to lane 9. Have the expansion, only one allele should show expansion. These are the two allele of 3, 4. There is an expansion, but there is no phenotype. But here you can say it's incomplete or reduced penetrance. The age is 16. This child is expected to develop the disease later in life because Huntington disease is a delayed onset disease. So do you understand the term, your problem set? Now, one more thing, please don't get lost in calculations in problem set. They are huge, but understand the concepts. They are very, very important for sure. How do we know each lane? Yes. How do we know each lane corresponds to the one? That's all. Lane 1 is for 1, 1. Lane 2 is for 1, 2, and so on. Lane 3 is for 2, 1. So you have to count the Roman numeral and the number. Each lane of gel only corresponds to one individual in the whole pedigree, right? You need to see the column and what is it corresponding to in the pedigree. Did I answer your question?
Yeah,
Everybody's
yeah, yes, you answered it. I just wanted to know, just in case, like, if we don't have the PCR like parallel to it, like, what if they were side by side? How would I know which
No, no,
lane
it has
will?
to be parallel. It has to be parallel.
Oh,
It's
okay.
said like that. And when you say somebody's asking, how do you know there's a phenotype? It is dark in color. If it is not shaded, now this you should know by now, please. Okay. Shaded is phenotype. Unshaded means unaffected, no phenotype, right? Does that make sense? Is everybody good? Yes, ma'am.
When we have a deceased patient, like we have in this genotype, we assume that during their time they did express the phenotype, correct? Like dad 1, 1, he is
Dad,
shaded, but he
yes, but the
deceased. So we assume, okay.
But the key is you have to understand that even if you have an expansion, you still have to express this disease at the age of 40 or around, right? So if you have at 60, that means it's written here, right? Onset 40s. So that's okay. If you have a genotype and you didn't express at an age, you have to watch the age. Inside the box, there's age is given. So this individual may develop the disease later, right? So not just the expansion, but also the phenotype and age. Age is also important. That's why delayed age of onset diseases individual may not show at an early age. That doesn't mean that they have incomplete penetrance. You have to wait for their right age and then decide whether it's incomplete penetrance. Does that make sense? Yes,
Yes,
everybody.
yes, yes.
Let's go further. So now look at this all. Yes, anybody has any questions? Which one was incomplete penetrance again? Okay, again, you're being your Zen. So I don't go back again and again, but that's okay. I'm just joking. So which one is incomplete penetrance is when you have a genotype, which is lane 6, corresponding to individual 2, 2. This is not patient, this is just analyzing pedigree. The which is incomplete penetrance is lane 6 when you see the expansion 40 and above and you don't see the phenotype. So it's 2, 2, which is showing incomplete penetrance. 2, 2. 2, 2. 40, yes, 40 and above should show the phenotype. Yes, yes. Everybody's good, right? Everybody's good. Did I answer you who's showing incomplete penetrance? Yes, this is your chance to ask question, clarify doubts. 3, 1, 3, 1, 3, 1 is okay. 3, 1 doesn't even have the expansion. It's lane 4. You have to have 40 and above to express the phenotype. So that's okay for lane 4, which is 3, 1, not to show the phenotype because he does not even have the genotype. Yes, absolutely, Aaron. Age is in the circle. Age is in the circle. Onset is written at the bottom. Expansion is down, right? Age is in the circle. Is 3-1 is if 3-1 is AA? Absolutely. If it's an autosomal dominant disorder and in autosomal dominant disorder, unaffected individual is little a, absolutely. 3-1 individual is little a. That is very important. First, you should know who is the mutant allele here. Mutant allele here is capital A. I hope everybody is following. I am not rushing, please. Sorry, but I think you have time, right? Is the space good? So I'm answering everybody's question. Everybody's good? Yeah? Yes? Let's, everybody? Great. So let's look at the incomplete penetrance. It is given here. Students are confused. How do you count it? It's given in percentage. Incomplete penetrance is all or none. It's either 100% or not. So here it is given. But see, then you have to draw a Punnett square. And they are asking for what? 3, 4, which is an unborn child and their parents. Here, first you have to draw a Punnett square to calculate the to include penetrance. This is the mating between capital A, and little a, little a. To draw a Punnett square between capital A, mom, and a, little a, dad, find out the offspring that can be affected, which in this case has already been given, and times the penetrance. So you have to draw the Punnett square. And this is actually the common mating. The common mating is only one parent is affected. Why? Because autosomal dominant disease are not common. But please know that there can be two affected parents. So whatever is given to you, you draw Punnett square accordingly. Does that make sense? Everybody? So the key is identify the right pattern, give the right mutant allele. Don't forget to know which genotype, what is the genotype of parent. Quickly draw the Punnett square. Please first do Punnett square. If you know the fact is autosomal in the complete, then we wouldn't be able to. Yeah, absolutely. They have to say either from pedigree or from example that this is autosomal dominant. But see, if penetrance is given to you, it's automatically dominant. So you don't have to worry then. You have to assume and don't just break your head with this. How do we, I just told you how to do we calculate. I can go back and tell you again. How do you calculate penetrance? Someone saying, for example, for 3, 4, you have to find out. And penetrance given is 80%. First, you have to draw the Punnett square between the parents, which in this case would be capital A, little a, mom, and little a, dad. You find out how many kids are affected or offsprings are affected times the penetrance. Please see, do your practice questions and your lecture questions. They will help you, but don't get lost in the calculations because remember, every lecture will be tested. Penetrance can be using the formula risk of inheritance genotype. Absolutely, but you have to draw Punnett square to know what is the probability the children will be affected, then times the penetrance given. Yes, you have rightly stated that. Did I answer your question? Yes, Erin, tell me.
Do you mind just going back to the last slide really quickly? I had a
Yes,
question on that.
please. Yeah.
So for me, like I kind of go about it through the thing on the left. So obviously it's affecting many generations. So I think dominant And then for me, like I don't see male to male transmission, so I would think like I would go straight to X-linked dominant. How do you know right away that
Mm.
there's incomplete
Now,
reduced
very good
penetrance?
question. First of all, do we need statistical data for you to calculate the penetrance for this? You don't need statistical data. Whatever is given penetrance, you just see that. Yes, I will explain you why it's not. First of all, this is mitochondrial or not. Is this mitochondrial? Is this pedigree mitochondrial?
No,
No. Is this recessive or dominant?
dominant.
Dominant. If it's dominant, there are only two options. Either it's X-linked dominant or autosomal dominant. Yes?
Yes.
In autosomal dominant, first of all, you said I didn't see male to male transmission. You can say that right by here, right? Because there might be sometimes male to male transmission and it still can be X-linked recessive. I'll tell you how. So you don't do that for pedigree. First you rule out mitochondrial, then you rule out it's recessive or dominant. So you're clear it's dominant now, right? Now there are only two options left, sorry. Either it's X-linked dominant or autosomal dominant. Now you tell me if you see these two phenotypes, how is it that it's X-linked dominant and mom is not even having, both should have the shaded area, right? Because there is maternal inheritance when you say X-linked. How is this that mom has nothing?
I see. Okay.
Yeah. So you have to
It
see it
had to
well.
have come from 2-4.
Exactly. So at least mom should have been shaded. You get my point? Otherwise, how would they
Yeah.
get the, yes, because there's no male to male transmission. If the sons are affected, if you're saying X-linked dominant, there's no male to male transmission, right? And mom is not showing anything.
That makes sense. Thank you.
Yeah, so you have to understand. And also, if you count the number, I think it's atypical, though it should be almost same, but you can clearly rule out X-linked dominant, please. Also know that X-linked dominant is rare, though only criteria for X-linked dominant. Okay, now, one more thing which is very, very important that you forgot is in X-linked dominant. So how do we ruled out it's not mitochondrial, we ruled out it's not recessive, we identified it's dominant. But tell me, how do you say it's an X-linked dominant pedigree? When what do you see? When do you call it an X-dominant pedigree? What do you see more than the other? No, males affected is X-linked
Daughters.
recessive. Sorry, daughters. Female affected more than male. Is that in this case? Are female affected more than male? Are daughters affected more than male in this pedigree?
No.
Aaron, so this can be X-linked dominant. So there is a criteria. Actually, if you would say, which is not a typical pedigree, it should have fitted in X-linked recessive, where male are more than female, but then it's not recessive. That's why it's absolutely autosomal dominant. Did I clear that?
Yes.
Um,
Thank you so much.
sorry.
Right? In X-linked dominant, daughters should be more affected than, or female should be more affected than male. Yes, anyone? Yeah, somebody has a question, Karan?
Dr. Bug, one question.
Yes.
So for this one, I understand like the probability that 3-4 will be affected. I understand where 0.5 comes from, but where do we get
It's
0.8 from in
given.
this pedigree?
It's given. 80% is given.
We were told that it's 80%.
Yes, you have to
Okay,
read the
got it.
question. You're given. It's given. It is given. The
Um,
80% is given. Yes.
I have another question. Sorry, I don't know how to raise hand, so
Sure,
um,
sure.
so quick question, like how can you tell that the previous pedigree is not like recessive? Because
There's no skipping of generation. It's
I see.
absolutely dominant. Yeah.
Okay, and then do we have to calculate like incomplete penetrance for like all autosomal dominants or when we see the
Income
to copulate.
Incomplete pattern is for dominant disorder. So if you see the pattern as dominant, an example as dominant, then you think about incomplete penetrance. Yes,
Okay.
incomplete penetrance is for dominant disorder. It's not talked in recessive disorder, no.
Okay.
Yes.
Thank you.
Can I take more questions later? Yes, tell me. Yeah, tell me quickly. Abdel, you have any question quickly? If you can tell me, yes. Is it Abdel? I'm
Yeah,
sorry.
can you hear me?
Yes, yes, yes. Tell me.
Yeah, what is if there's no skipping generation and then we have two unaffected parents and they have like someone affected? Is that like reduce penetration autosomal dominant?
See, reduced penetrance can only be assessed. It cannot be assessed alone by pedigree when you just see skip of generation in a dominant pattern. That's all. You can confirm that unless you run a gel and know it. Does that make sense? If
Yeah,
it's
yeah.
a dominant pattern, there should be no skipping, but there may be some skipping, maybe one or two. And also understand when you count the whole. But when you see is it vertical or horizontal pattern, you don't have to see both sides. Only one side is enough. Does that make sense? Either this side should be dominant, having vertical pattern, or this side. But you're very right. Only one skipping, that means could be incomplete penetrance. That's all. Nothing else you can know otherwise from the pedigree, except skip.
But we cannot just confirm from just the pedigree, right? If it's incomplete penetrance.
If it's incomplete penetrance, how will you confirm it from pedigrees? You will see a skip in a dominant pattern, which you should not be seeing.
Okay, yeah. Thank
Got
you.
it? But those
Yeah.
skips should be very, very scanty, please. They can be skipped like recessive, okay? But otherwise, it's not concluded from pedigree just by looking at the skip that you need more information. Skip has to be a lot, Alan, to call it recessive. What I'm saying is, so please, please, and please, right? First of all, if you don't concentrate on one pedigree and clarify your concept and jump to one and the other, I'm telling you, you'll be thoroughly confused. Right now, we are talking of autosomal dominant. I'm not talking recessive. So when I say autosomal dominant and you see one or two skip, that may be because of incomplete penetrance. Please don't bring recessive in it. When it is recessive, I'll show you the pedigree, which looks nothing like dominant, and it's not with one or two skip. Okay, can we please stick to dominant and not bring recessive inside, right? Otherwise, students will get a wrong information. Alan, I would address it later, please. Is that clear, everyone? I will tell you, recessive is a skip, but not one or two skip. It's an entire skip with a horizontal pattern. You don't see this pattern in recessive. Right, everybody? One skip is not recessive. Are you clear, Alan, please? One skip or two skip is not recessive. Yes, I'll show you what is recessive. Is everybody good? Let's stick to questions around one pedigree so that we can then do autosomal recessive and you will get more and more clear. Does that make sense? Yes? Please? I want you to get a clear message. I don't want to confuse the students, right? Yes, penetrance is always given. Yes, absolutely. Penetrance should be given to you. You can't assume penetrance. It's all or none. Absolutely. Right? Now let's go further. Okay, great. So let's see this now. Definitely, this is no skipping of generation. This is autosomal dominant. All I want, I'm not going through every question, but bringing your attention. This is the mating between an affected parent for autosomal dominant and an unaffected parent. This is a common mating because autosomal dominant diseases are not common. Only one parent is affected, but it is possible that both parents are affected. What you should not make a mistake, number one, is identifying the pedigree. Number two, which is the mutant allele. Number three, what are the genotypes? If they're saying there's a mating between an affected parent and an unaffected parent, then drawing Punnett square, you can quickly do it. Does that make sense? Right? Yes, you don't add half for any autosomal dominant disorder unless they ask sex, please no. Even for X-linked recessive, which is sex biased, I will show you, you don't need to add another half if you draw Punnett square. I'll encourage you to draw Punnett square for any pedigree given. For any, sorry, pattern given. Giving from pedigree can be you might make a mistake. Again, autosomal dominant clearly, but as I told you, one questions you can learn 5 concepts. Please don't forget to rule out pedigree of each and every example. For example, what is fragile X? Anyone? Fragile X is? What is fragile X? Yes,
X-linked
it's
dominant.
X-linked dominant. Absolutely, right? Let's stick to one inheritance pattern at the same time. And then we talk about now comes autosomal recessive. Yes, Erica, tell me.
Hi, Dr. Bach. I just had a quick question about the autosomal dominant.
Mm-hmm.
So if we were to ever be given two affected parents who would
Yes,
be
yes,
big A, little a for both of them,
absolutely.
and
100%.
one of the offspring were big A, would we just eliminate that since it's
No,
legal?
no, no, no. Please don't do that. Please
Okay,
don't
so
do
still
that.
include it.
Yes, you will include it in the Punnett square, but you are, unless they tell you, but you don't know. You have to count them, please.
Okay, so they would just be deemed affected as with the other big A,
Yes,
little a's. Okay.
absolutely. Please don't eliminate them in the Punnett square. No, please don't.
Okay.
Yep.
Thank you.
X-linked dominant, right? Good question. Good question. Keep asking question, but stick to one inheritance pattern, right? Otherwise, I don't want to confuse the students. Autosomal recessive, it's same like autosomal dominant, male, female equally affected, male and female equal chance. There is male to male transmission whenever it is autosomal dominant. And in autosomal recessive, the key is you should know the key characteristic difference. It is not possible that both parents do not carry the mutant allele. They have to carry the mutant allele. Don't forget the mutant allele is here little a, and this is the mating which is more common mating between two carrier parents. Please, please, and please, it's not possible in autosomal recessive that both don't carry the mutant allele which is little a. In autosomal recessive disorders, parents and children are carriers. They are obligate carriers who are given by dot. Those which are half line, they are given by testing. Yes, you have a question, anyone? Anyone has a question? Question, great. Let's look at the autosomal recessive pedigree when yes, Jasmine.
So, would 2, 3 then be the proband if they got the testing, to determine their genotype?
2, 3, 2, 2, 3. There's no genotype here. There is this pedigree as dot shows they are carriers, as half line shows they are carrier. But this is carrier by testing. This is carrier by pedigree.
Got
Okay?
it. Thank you.
Yes. Yes. Very good. Half shaded box is also carrier, which is your half shaded box is also carrier, but not by pedigree, but testing, which is 2, 3. They are carriers too. Did I clear that, everyone? Okay. Yes, let's go further. So did I tell you male and female equally affected? Both parents need to carry the mutant allele. There is male to male transmission, please, in autosomal dominant or recessive or any autosomal disorder. And then comes the 2/3 rule. 2/3 rule applies in 3 conditions only. When the disease inheritance pattern is autosomal recessive, both parents are carriers, and the child they are asking or sibling they are asking should be unaffected. Then only 2/3 rule applies. If these 3 things are missing, please do not apply the 2/3 rule, which is both parents carriers, autosomal recessive disease, and the individual they are asking unaffected. Info acquired from pedigree versus genetic. Yes, very correct. When you say obligate carriers, they are from pedigree info. And when you say genetic testing, yes, that is the half shaded. Yes, absolutely. That's why when you do a question, you keep going back until you see an obligate carrier. Know that in two-third rule, you do not count little a, little a. Don't do it in a clinical presentation for inheritance pattern, please. Just the disease and the gene. Yes, anyone has a question? Okay, so can we use two-third rule? No, you don't use two-third rule for dominant disorder. No, you don't. No. Two-third rule is only for autosomal recessive. Both parents have to be carrier and child should be unaffected. I think normal, yes, normal for autosomal recessive, yes, yes, is capital A, capital A, absolutely. No, not otherwise they would be carriers. If they are carriers for autosomal recessive, it should be capital A, little a. If they are disease, it should be little a, little a. Is math different for carrier, dot, and yes, there's no maths. It's just testing. When this is half shaded, it was found out by testing. If it is a dot in the middle, it was found out from pedigree. The way it was found out, it's no maths here. Yes?
No, I'm talking about when we calculate, like, if they ask the probability of like a child having it when we go back on the, do you mind going back to the pedigree? I'm still talking about autosomal recessive, by the way, not not hopping.
No, I don't mind that. Only thing is I just want to give the key points. And yes, yes, tell me.
No, back where it had the half and the dots, that pedigree. Because that's why I was wondering if the math is different.
Please,
Yeah,
yeah.
yeah. When we do the math, say if there was 3-2, there's no dot, we don't know. And we're doing the math going back. Or if we do, if 2-3 has a child with a normal woman
So
or
see, let's not do if and but, please, right? Let's not do if and but here. So this has a child, but now you can assume anything. Does that make sense? It's not showing in the pedigree. I think do the same when you are doing questions, right? Stick to what is on the slide. So you are telling me now they have a kids, but it's not even shown in the pedigree. So
no,
why
I'm
are
saying so, but if no matter how the carrier was found out, provided on the pedigree, do we do the math the same? That's pretty much what I'm asking.
Oh, absolutely. That is no
Yeah.
doubt. Yeah, that is no doubt that it doesn't matter that you found out pedigree, you found out carrier from the pedigree or by calculation. They are still carriers, but it's that it's just a different name given to them. These
Oh, okay. Sorry, my
these are
English is not
no
good.
worries, no worries. These are obligate carriers in autosomal recessive. If you have an affected child, it is not possible that both don't have the mutant allele. Since they both are not shaded, they both are carriers. So that is why they are obligate carriers. That means they have to be carrier according to pedigree. So you give them a little dot. But look at this one. You cannot say neither you see their children, other spouse. So this is why this can be determined that this is obligate carrier. So this was determined by testing. Does that make sense?
Yes, thank you.
Yes. Is the same probability one? Is this the same probability 1, 1, and 2, 3 to pass the mutant allele? Where are you, please? Are you in this slide? Can we not go back, forth, back, forth? So if I'll ask everyone, this slide is done. Which slide are you asking me, please, Alan? Is it this one? Yeah, this one. Yeah, tell me. So what is the same probability 1, 1, and 2, 3? Yeah, it is the same. They don't behave differently. They are just named differently, please. One is carrier by testing and one is obligate carrier. The parents are obligate carrier and 2, 3 was just confirmed carrier by testing. That doesn't mean they are behaving as different carriers. Their carrier passing, everything is still the same. Did I answer you? Yes. Anyone has question on this slide, please? Are you clear with the two-third rule? Okay. Anyone has question on this? You need to know disease and gene. Now look at this pedigree. Somebody was asking me when I said that there is skipping of generation In autosomal dominant, there's only one or two skips. Somebody said, oh, but the skipping is also in autosomal recessive. Now look at this skipping. Is this abnormal? This is a lot of skipping, and there's an horizontal pattern. So this is a recessive pedigree. Somebody asked me why it's not X-linked recessive, because you see male more than female. Yes. So why is it not X-linked recessive? First of all, are daughters affected in X-linked recessive? Who is affected in X-linked recessive, daughters or son?
Sons.
Sons, but here, can daughter be affected in X-linked recessive? Yes, she can be, maybe, but in this case, it cannot be X-linked recessive because at least both should have the mutant X. Father also and mother also. So that is why daughters are only affected. Yes, daughters are not affected usually in X-linked recessive unless both parents have the mutant X. Very correct. But they may be affected. So this is not, somebody asked me and they are confused that is this X-linked recessive because you see 2/1. First of all, this is not a typical autosomal recessive pattern. In autosomal recessive, they should be almost equal. You see more male, but still it can be X-linked recessive because both parents are not shaded. This is not possible if daughter is affected in X-linked recessive. So this is an autosomal recessive, male, female, almost equally affected, skipping of generation, and so common mating between two carrier parents, right? Now, one thing I want to tell you, if they are just asking carrier, for example, they asked about 2, 3. What is the probability? Let's do 2, 3. And I'm not doing the whole pedigree, but bringing your attention to some concepts. They are saying, let's go to B. If 2, 3 marries a normal female and they're asking about carrier. When they're asking about carrier, then you don't have to worry whether the female has what genotype. It's just asking you if this guy, this father marries another normal female, what is the chance this will pass mutant allele? For any parent to pass mutant allele to next generation in autosomal recessive is half only because carriers only have one little a. Does normal mean heterozygote dominant in this case? First of all, you should not use the word dominant in this pedigree. Okay, so sorry. So you're saying, does normal means homozygous? Absolutely. But what homozygous? Capital A, or capital A, little a? So in autosomal recessive, when you say normal, yes, it should be. One thing is, let me bring to your attention. In autosomal recessive, there are two normals, carriers and absolutely normal. Carriers are also normal. Carriers are phenotypically normal. So when you say phenotypically normal, it can be carriers and normal individuals. But carriers are not genotypically normal. So yes, if you're saying absolutely normal, not talking about carriers, in autosomal recessive, it should be capital A, capital A. Yes, you're right. 3 is X-linked recessive, right? Just a second, am I making any error? 3 is not X-linked recessive. It has to be autosomal recessive. 3 is not X-linked recessive. This is recessive, but this is not X-linked. This is autosomal recessive pattern, which I'm showing right now. Could you explain? Okay, yes. Could you explain why 3, 5, and 3, 3 are expressing the mutation if this is recessive? 3, 5, and 3, 3. You tell me why. There comes obligate carriers. How is 3, 5, and 3, 3 manifesting disorder? Yes, because both parents are obligate carriers. It cannot be X-linked recessive because the dad would have to. Yes, this cannot be X-linked recessive because in X-linked recessive, daughters are not affected. If daughters are affected, both parents should have the mutant X. Yes, that is why it's not X-linked recessive. But why 3, 3 and 3, 5 are affected? Because 2, 4 and 2, 5 are obligate carriers. Draw a Punnett square between two carriers. There is 25% chance children will be affected. That is more common. That is here. They are more than 25. But yes, they are affected because both parents are carrier. Did I answer you? Everyone is good with this slide? Any question? Great, let's go further. So this is autosomal recessive, pedigree 3. It's not X-linked recessive. I told you the reasons why. So in autosomal disorders, male and female transmission is possible. They are almost equally affected, and both parents have equal chance to pass the mutant allele. When it's X-linked recessive, please know that both parents need to carry the mutant allele, unlike X-linked dominant. Whenever you see double line, please don't jump to autosomal recessive. You are very sure it's recessive, then rule out, is it autosomal recessive or X-linked recessive? Is male and female equally affected here? Are male and female equally affected here? So this is autosomal recessive. Yes, this is autosomal recessive pedigree. Let me see if I want to bring any other point to attention here. Okay, what is the probability for, okay, what is the probability for 2 is a carrier? So first you have to find out, is this autosomal recessive? On the exam, will they clarify whether I mentioned genotype? In the exam, Karan, they should clarify that you assume that they are normal and not affected, but you still have to see the pattern. For example, they are saying that for autosomal recessive, even if they are saying that the two parents are phenotypically normal and you see affected child, it has to be carrier. You have to see it from the pedigree too. So what is the probability for 2 is a carrier? Somebody saying half, somebody saying 4, 1/4, 4, 2. Who's going wrong here? Yes, 2/3. I'm not asking affected, I'm asking carrier. And this individual is phenotypically, sibling is phenotypically unaffected. So 4/2 is 2/3. First of all, who's saying 1/4? Please go back and draw a Punnett square between two carrier parents. For autosomal recessive, you're wrong. Who's saying half is not wrong, but this is 2/3 rule, because the individual they are asking is unaffected. Yes, absolutely, Ariana. 4/2 is unaffected, so carrier probability instead of half will become 2/3. Got it? Yes, let's go further. If you have any more question, I can talk about them. Again, see, you are saying skipping of generation, but there's a lot of skipping of generation in autosomal recessive. This is null marriage. This is not consanguineous marriage, or this is not related individual. It looks like a heart, but you see broken hearts here. There's a null marriage. Yes, it looks like a heart. I'm just joking. So now, so what is this? What is this pedigree? This is absolutely autosomal recessive. Let's do one, which is, what is the chance for 1 is a carrier and for 2 is a carrier? This is what I was telling you when you say obligate carrier. You cannot say from both these parents what is their carrier probability. So you go up until you find obligate carriers. Now probability 3 is a carrier is 2/3. Pass to the down half, another half. So probability 5, sorry, 4, 1 is a carrier is 1/6. Mom is a carrier is 1/6. Let's go to this one. Again, by this little pedigree, you cannot say the carrier probability. You go back up. This is affected, so mom has to be carrier. Pass the mutant allele half, another half, 1/4. Probability mom is affected, 1/6. Probability dad is affected, 1/4. Probability child is affected when both are carriers. So somebody did that for me. Very good, 1/6, very good. So probability the child is affected will be 1/6 times 1/4 times 1/4. Absolutely. Please don't get lost in the calculation concept. When do you need to get 2/3? Why do you want to pass? Because the ability for a parent, carrier parent, to pass the mutant allele is only half. In autosomal recessive, they are only carrying one mutant allele, right? Again, double line, this has to be recessive. And is this autosomal recessive or X-linked recessive? Anyone? Is male and female equally affected? Yes, male and female equally are affected, so it's autosomal recessive. Now, just to bring to your attention, because you keep drawing Punnett square in autosomal recessive between two carrier parents, and in their genotype would be capital A, little a, and capital A, little a. But look at this. Tell me, what is this mating between if it's an autosomal recessive disorder? But first, what you did is identify the disorder. It's autosomal recessive. This is the mating between little a, and yeah, somebody said capital A, little a is wrong. So mom is little a, and dad is capital A, because you don't see any phenotype here. You cannot make the dad a carrier. You can say there is somebody who's saying only females affected, though this is not X-linked. This is autosomal recessive. I'm talking. There's no sex bias here. There is male to male transmission. There is no sex bias here. So this is the mating between little a, and capital A, capital A. Does that make sense? So do the other matings and in inheritance and draw the Punnett square. Right, let's go further. There is no female, only females affected though. Where is it only female affected here in this pedigree? Oh, you mean to say as a number? But again, do the same criteria. If this daughter was affected, do you think if it was X-linked recessive, none of them had the mutant X? Amir is right that females are only affected, but is it X-linked recessive? Because okay. Let me go back again, Amir. Let me go back again. When you're saying females are more affected than males, what is that pedigree according to you, where females are more affected? No. Let me answer stepwise, please. X-linked dominant. Is it dominant pedigree? Is it dominant or recessive? So it's gone. Don't think about it, right? Did I answer your question? Now I have, you just seeing female affected. Remember, female affected is as X-linked dominant. We assume Y. What is Y linked? Why is somebody saying Y? I don't get that. Please, can you unmute and ask me? Okay. Where is the Y coming from, please? Are you assuming 3-4? Somebody saying, are you assuming 3-4 is genotypically normal? 3-4 is genotypically normal. No, I cannot say that. 3-4, yes, I can say about 3-4 because from the pedigree, I know that this is the carrier. Yeah, I'm assuming 3-4 is genotypically normal. Did I answer you? And very good. Somebody has pointed out, but no, he's just asking, is 3-4 genotypically normal? I know it's not part of the family. That's okay. But 3-4, somebody's asking, is he genotypically normal? Yes. Uh, there are too many questions here, believe me. Are you still here on this pedigree? So it will be capital A, yes. 3-4 would be capital. 3-4 would be capital A, yes. Yes. 3-4 will be genotypically normal and will be capital A, absolutely. Did you get this? So somebody was saying you see more females. No worries, but more females is X-linked dominant. So this is not a dominant pedigree. You know that two carrier parents affected male 1/4. 2/3 rule, if they are saying that the individual or sibling they're asking carrier probability is unaffected, right? X-linked recessive. X-linked recessive. Now, do you see X-linked recessive pedigree clearly here? Skipping of generation by female, male more affected. So if one kids were affected, it would be AA. Yes. If any of the children, let's complete the questions in the slide because this will confuse the students, please. Okay. Do you want so yeah, let's hold question till the end. Let me finish this. I think that's a better idea, please. Please write down your question and I'll give you 15 minutes because I think it will confuse the students, Please, okay? From now, let me finish the pedigree. So everyone is clear till now. In the last pedigree, there were more females. Still, More females is seen in X-linked dominant affected, and it was not a dominant pedigree, so it was autosomal recessive. And that's the only pedigree it fits in. In autosomal recessive, both parents have to be carrier. Although you should know common mating in autosomal recessive, mating between two carrier parents, but it's not uncommon to do or draw a Punnett square in autosomal recessive between a affected parent and a carrier parent if children are affected, right? X-linked recessive. In X-linked recessive, there is no male-to-male transmission, skipping of generation by female, male more affected than female, clearly, or X-linked recessive. In X-linked recessive, males are not carriers, males are affected, only females are carrier. Please identify the pedigree, give the mutant X, please. Common mating is between the carrier mom, which is in red, and unaffected dad, an affected dad, and an unaffected mom. This is common mating. But there could be other possible combination too. Male are not carriers, they are hemizygous, because if male in X-linked recessive even have one mutant X, they will express the phenotype. Yes, the royal family hemophilia, yes, nothing wrong in that, right? Don't draw it and waste your time though, okay? That's all I would say. So X-linked recessive, no male-to-male transmission. Also, one more thing, please. In X-linked recessive, only moms and daughters are carrier. Unlike autosomal recessive, parents and children are carrier. Very important. No male-to-male transmission. Know the examples for autosomal recessive, X-linked recessive, autosomal dominant, and mitochondrial all. So you did X-linked recessive. One parent can be carrier, yes. Who are carriers? Mom and daughters. There's no male-to-male transmissions. Male are never carriers. Male are hemizygous. If they have a mutant X, they will express the disease. Know the disease and gene. Manifesting heterozygous just for X-linked disorder only when X-linked inactivation is cued. Sometimes carriers become symptomatic. That's why they are known as manifesting heterozygote. Is this clearly an X-linked recessive pattern? Absolutely. Skipping of generation, so recessive. And male more than female, right? What is the probability that 4-1 is a carrier? Is there any probability 4-1 is a carrier? Anyone? It's X-linked recessive. Is there a probability that 4-1 is a carrier? Do you think there's a chance? Absolutely not. Males are not carrier. Male and male transmission is not possible. Females are carrier. Moms are carriers. Daughters are carrier. What is the probability now 4-4 is a carrier? Though you can do it from pedigree, I will encourage you to draw a Punnett square. I will just do this one, which will be nice to explain you. So if you want to find out carrier probability of 4-4, you have to go back. Why? There's nothing here that you can find out from immediate parents. So if son is affected, is 2-3 an obligate carrier and she's female? Is mama carrier? Is mama carrier? So mom is a carrier and the probability she will pass the mutant allele to a mutant X to the daughter is half. So carrier probability of 3-7 mom is half. Males are not carrier. So this should be the mating between a carrier mom and an unaffected dad. Either you draw a Punnett square, which is on the left. I'll show you. Either you draw a Punnett square, which is on the left between carrier mom and an unaffected dad, but don't forget to carry over the carrier probability of mom, which is half. Also, please, a common mistake students make when a carrier mom in X-linked recessive marries an unaffected dad, 50% daughters are carrier, 50% son are affected. But if they don't ask daughters and carriers, they ask offspring. Then it is 1/4, and that is a male. So please don't assume sons and daughter are different from children or offspring affected. So when a carrier mom, X-linked recessive, marries an unaffected dad, the children affected is 1/4. But here, don't forget to, yes, carry on the carrier probability of mom here, which is half I've drawn in the box. If you want to do from pedigree, which some students like, okay, this is the carrier mom. Probability she passed the mutant X is half. Probably she will pass the mutant X to the to 4 is another half, but don't forget to add in pedigree another half that it's a male, because only males are affected. So that is why doing from pedigree, which is right, I've given you one option rather than doing from Punnett square. Please stick to Punnett square, because in Punnett square you don't have to give another half. You know that this is a male. Does that make sense? Everybody is good, right? Again, this is X-linked recessive. You see male more than female. Now, this is very important for you to understand that in X-linked recessive, daughters are not affected. But when daughters are affected, when both have the mutant X. So in this case, is it obligate for 3 to be a carrier? Is it obligate for 3 to be a carrier in this case? Absolutely. So this is a mating between, when you see this family, this would be a mating between a carrier mom and an affected dad. Does that make sense? Which is in your 4-5. Sorry, I'm just telling you that if you see an affected daughter, that means both are carrying the mutant allele. So it's 100% that 3-3, which is here in the question, sorry, C, 3-3 is a carrier, 100%. Does that make sense? Yes? 3-3 is a carrier. Yeah. And due to X inactivation, there can be abnormal pattern, but let's not do X inactivation detail here. Right now, assume there is no skewed X inactivation, right? So you understand? And also, please see here, this is a mating between an affected mom and what is the dad? So dad is normal. Is dad totally unaffected? Yes, because you don't see anything wrong with the children. If the daughter was affected, you would assume that the dad is. And anyways, if dad had the mutant X, would it show phenotype? Would it be shaded? In X-linked recessive, if dad are hemizygous, right? If they have even one mutant X, they would express the phenotype. So here you have to assume that the dad is, sorry, not dog. So the dad is phenotypically and genotypically normal, and mom is X-linked affected, right? Does that make sense? Let's go further. One more thing, please. On the right side is E. E is the pedigree between an affected mom and an unaffected dad. So just see what is given. Though this is not common mating, this is a mating between affected mom, 4-5, and an unaffected dad. The probability will change, right? Is this X-linked recessive? Absolutely. Male more than female, skipping generation by female. When a carrier female marries an unaffected dad, 1/4 children will be affected. When a carrier mom marries an unaffected dad in X-linked recessive, 1/4 children will be affected. Here, it's very important for you to understand that she is affected only because she got mutant X from both. The boy got the mutant X only from mom. There is no male-to-male transmission in X-linked. And the daughter is a carrier, got a mutant X from the dad. From both, from mom, from dad. Go backwards and differentiate, right? X-linked dominant, only and only criteria is you see female affected more than male. Daughters become affected in X-linked dominant disorder, that's all. Mitochondrial, mom affected, all children affected. Dad affected, none of the children affected. Heteroplasmy is for mitochondrial, and depending on the mutant and normal mitochondria and the expression threshold, the phenotype will be expressed. Know the example mitochondrial, LHON, and MERF, and MELAS. Totally mitochondrial, mom affected, all affected. Mom affected, all children affected, right? Does that make sense? They're asking about the brother, sorry here. Brother definitely 100% inherited from mom. Mitochondrial is maternal. If you see this pedigree, which is for 3-9, if 3-9 marries, well, if dad is affected, none of the children are affected. That is mitochondrial. And why? I want to tell you why. Why linked? Please don't be confused. It should be father, son, father, son. That is why. It's not male more than female. It has to be a pattern of father, son, father, son. This is for inheritance. Well, give me 5 minutes. I will then tell you, because I need a break, please. I will tell you just quickly do the replication and repair, right? Just take a 5 minutes break. I just need another half an hour, please. Then I'll open for question. Is that okay, everyone? Take a 5 minutes break, please. I will open question at 11:30. If you want to stay, you can stay on. I will do replication, repair, review after 5 minutes. Are you getting ready? Let me start the replication repair in a few. Let's see what we have for replication or repair. Is everyone ready? What I'm going to do in replication and repair, I'm going to just point out. This is this in 15 minutes. Then it's open for question, please. Is everyone ready? Okay, let's do replication and repair. Some people might have missed the recap. Let's bring your attention. I'm not teaching here. I'll just tell you in 15 minutes what is important. Then it's open for question. I have put these pointers. Please, they are aligned with learning objective. If you want to read them and benefit, it's up to you. What you need to start with is nucleic acid. They are polymer of nucleotide made up of base, sugar, and phosphate. You must understand your bases and how do they differ in group, because there is an endogenous mechanism of DNA damage that can modify these bases. That's why you should know which bases have what group. We identify adenine, guanine, and cytosine have amino group that can be given up. Minor bases are not taught here, but yes, you see them in conversion. For example, adenine changes into hypoxanthine, guanine into xanthine. Only there you saw them. If the base is modified, it will be repair, which will come next, base excision repair. Similarly, you should know they are not purines, but they are pyrimidines, single ring, and have different groups. We identified that amino group out of all of them is with cytosine, right? We did identify groups because there are endogenous agents of damage. One of them is deamination, and we talked about them. Then how they are stacked and different bonds. Please know that 3' 5' diester bond is always on the same strand, not on the opposite strand. Opposite strand is hydrogen bond, right? This is phosphodiester bond. One more thing is students are confused with CpG islands. CpG islands are those CG repeats on a single strand near the 5' site. CpG islands we have not taught you in detail, but we also taught you that near 5' end is promoter. Later on, we will tell you when out of CpG, the cytosine, if modified by methylation, what does that result in? We have not discussed that in detail right now. But modification of cytosine of CpG is part of DNA and not histone. So don't confuse the both. So know the different bond in between two nucleotide is 3' 5' phosphodiester bond. Between the base and the sugar is glycosidic bond, because later on you will have enzyme glycosylase for base excision repair, and you will have ligase, which brings these nucleotides together with the help of 3' 5' phosphodiester bond. You need to understand that in between two bases, there is hydrogen bond. Stronger the bond, more energy you need to break the bond. Polymerization is always 5' to 3'. What happens is that one strand is in 5' direction, the other strand is complementary and anti-parallel. You need this 3O to be free to form the 3' 5' phosphodiester bond. In between two strand is the hydrogen bond. CpG islands are on the single strand. Modification of CpG island is not same as modification of histones. Does that make sense? We have done replication, not transcription or translation, but you must understand that this double-stranded DNA is on which you do replication. 5' is proximal promoter, and it is always on the 5' side, and it is usually near that there are CpG islands. CG repeats. Modification, we have not taught you in detail. We will do that later. Should you know repetitive classification and their significance, whatever is given in Dr. Saniviratne's slide, you must distinguish. Why? Because satellite repeats and their instability is addressed in Lynch syndrome. So whatever repeats and their concept or clinical in conditions have been addressed by the PowerPoint. In the PowerPoint, please do that. I address the CG repeats, which are not on double strand. They are on the same strand, bound together by 3' 5' phosphodiester bond. Then comes replication. In replication, we will understand polarity. I told you to know repeat. Differences, bring differences wherever you can, whatever is in the slides. Don't forget not to read them. Histones and nucleic acid concept was clearly explained. Histone is positively charged and nucleic acid is negatively charged. These modification, please and please and please, are not for DNA. DNA modification have not been taught to you yet, which was the CpG. This modification is for histone, most common acetylation. We have explained you euchromatin versus heterochromatin well, which is transcriptionally active, Euchromatin is the outcome of histone acetylation, not deacetylation. That distinction you should make. When you do replication versus repair, only and only thing please do for me, keep the right enzyme, right protein in the right process. Don't mismatch there, right? Also know the function of each and how they are important in replication. For example, topoisomerase 1 versus 2, who induces negative supercoil, 1 or 2? Which one induces negative supercoil? 1 or 2? 1 or 2? Is it both or is it 2? 2 induces negative supercoils. No, for example, what is single-stranded binding protein doing? Preventing it from re-annealing. So not just the protein and enzyme, its function. Know what is polarity. What is 5' to 3'? Also, do not forget to bring in your drugs and clinical condition along with these enzymes and their defect. Please know everything around the replication fork. Yes, please. Anybody has question? Polymerization, yes. Also, some students are confused what is in 3' to 5' direction. Polymerization is always 5', 3'. Poof reading is backward 3' to 5'. But what else moves from 3' to 5' direction is your DNA polymerase. DNA polymerase in eukaryotes is alpha, beta, gamma, delta, and epsilon. Know them all. Know them all, please. Right? DNA polymerase moves in 3' to 5', but polymerizes 5' to 3'. Are both strand acting as template? Absolutely. This is not transcription. Both of them act as a template and are known as parent strand. You can call them template and replicating strand, template and replicating strand. It's on you. Know the polarity. What is in 5' to 3'? What is 3' to 5'? Know the replication fork. Know that this is bidirectional. Bidirectional. Don't get confused. Learn them halfway. Just draw a line here first. First find out what is leading and lagging on the left, then find out what is leading and lagging on the right, and you will understand where are the Okazaki fragments, new or old. Know all of them. Which one is for mitochondrial? Which polymerase is mitochondrial? Anyone? Mitochondrial is gamma. So you have to know your polymerase. Now, this is eukaryotic replication that I'm addressing. Please bring in difference between pro and U, which is given in table by Dr. Senivratne and Dr. Panini. Gamma is mitochondrial. Absolutely. No function of each enzyme, what they are doing. Absolutely. Bring them. Bring the polarity. Bring the isomerase. Bring their drugs, whether it's topoisomerase 1 or 2, pro or U, drugs. For example, cans, all the cans are anti-cancer and they inhibit topoisomerase 1 or 2. All the cans. Cans inhibit topoisomerase 1, pro or eukaryotes. Topitacan, I'm just making it cans. Topitacan. Eukaryote topoisomerase. Is it 1 or 2? Am I missing? Topitacan is topoisomerase 1 inhibitor. Yeah, absolutely. Yes. Eukaryotic topoisomerase 1 inhibitor. Okay. So don't forget to bring in your drugs for sure. Yes. Topoisomerase 1 can. I call them cans. You can call them anything. Topitacan. Topitacan. It's a can, right? Just to know them quick. Yes. But they are topitacan and they have their names, right? So eukaryotic topoisomerase inhibitor 1 is can. Bring in the differences wherever you can, please. Pro as well as eukaryotes, but know first the normal, first know normal to know abnormal. First know what is topoisomerase function, how they are difference between 1 and 2. Then bring in 1 is pro versus U. Then their drugs and examples, very important. End replication problem is at the 5' end of the daughter. 3' of the parent is elongated by enzyme telomerase using an RNA component known as TERC. Don't forget to bring in your drugs wherever you can. Telomerase is a reverse transcriptase because it uses of RNA to elongate 5' end of the parent strand. Don't forget to bring in the drug. I want to show you here that reverse transcriptase is a polymerase, is a reverse transcriptase, is a RNA-dependent DNA polymerase and elongates the tag sequence on the 3' end of the parent. 3' of the parent. Would you mind showing the prokaryotic slide again? I can know what you meant by. Yes, prokaryotic and eukaryotic slide, though this is the old one. You could see it's from Dr. Sani Viratne too. But I like the old slides because they have they're colored, but he has the same information, please. For end replication. Okay, what is end replication problem? End replication problem is that at the 5' of the daughter strand, when FEN1 removes the primer, eukaryotes, and first of all, end replication problem is only in eukaryotes, and it is at the 5' end of the daughter strand when the FEN1 can remove the primer, but polymerase cannot replace the nucleotides. So that genetic information is not lost. What nature has done is telomerase kicked in, elongated the 3' end by using an RNA component. And then RNA component was removed and daughter strand is also polymerized. So there's a chunk of nuclear material in front. So even after replication, you lose genetic information. The important in the middle is still retained. Any cell that reaches its telomeric threshold should go permanently into Z0 phase. Primer or template? What do you mean by primer on template? Can you please unmute and ask me so it can be quick? Yes, Amir, you can unmute. Will primer be on the template strand? No, this is not the primer of replication. Here you're not talking primer, please. Primer is restricted to replication only. In replication, you need a primer. You need the template strand. Here, it's not primer. Here, there is a problem at the 5' end. So what is reverse transcriptase doing? Using an RNA component sequence at the 5', it elongates the 3' sequence, which is tag. So there is no primer here, please. The primer, yes, is here. I'll show you in the next slide. This is the sequence of the parent strand that has to be elongated. So you will use a complementary primer RNA here, and the telomerase will elongate the sequence. RNA component, sorry, I don't want to call it a primer. It's not a primer. Did I answer your question, Amir? This RNA component, yes, TERC come first, absolutely. This should not be mentioned first. TERC comes first, then tag sequence is polymerized, then TERC is removed, and the 5' of the daughter is also polymerized accordingly, complementary to the parent strand. Yes. But there is no primer here. Primer is used in replication. It is the RNA component. Yes, no, I'm absolutely happy to answer. I will take more questions. Just let me finish the important concept. 11:30, I'll open the question and you can then make me go back and forth because otherwise students are confused. But if you have a quick question, yeah. Don't forget to bring, this is the sequence in the parent strand, 3' that has to be polymerized, and this is the RNA component used by reverse transcriptase. Don't forget to bring in your drugs, please. You know DNA polymerase, you know primase, and you know reverse transcriptase. Mitochondrial DNA is maternal. mostly transfer RNA encoding. That's why you see the transfer RNA mutation. Actually, they don't have introns, they don't have histones, they do not have a good repair. That's why they are more prone to mutation. Please do not forget to bring in LHON, MERF, and MELAS. For example, ragged fibers you will see in which mitochondrial disorder? Ragged fibers. Ragged fiber is MERF. Maternal, mother has the disorder, all children. Father has the mitochondrial disorder, none of the children. Heteroplasmy, yes, absolutely, is also addressed with maternal disorder. Repair. Repair cell cycle is only important to understand the role of P53. P53 kicks in whenever there's a DNA damage. Who does the DNA damage? Agents. Whenever there is a DNA damage, P53 kicks in, stops the cell cycle, allows the repair enzyme to repair, and if repair not happen, induces apoptosis. P53 is affected in ataxia telangiectasia. P53 is an important protein, but for it to be active, it has to be phosphorylated. So yes, P53 is important as far as cell cycle because that kicks in only or get activated only when there is DNA damage. DNA damage, be mindful of the common endogenous as well as exogenous agents. For example, only UV rays cause thymine dimer. It's only UV rays that cause thymine dimer, not ionizing radiation. We also discussed wall deamination, methylation, and oxidation here. We did replication error. Replication errors are seen where there are more repeats and when your proofreading has failed. Proofreading is what? 3', 5' exonuclease activity. Proofreading takes care that even when there are a lot of repeats, there is no replication error, there's no mismatch. Even if there is a mismatch repair enzyme can kick in. Know that these are the protein or enzymes involved: helicase, exo, endo, poly, and ligase. But what you need to differentiate is single-stranded from double-stranded. Don't mismatch their clinical condition, please. How is single-stranded different from double-stranded? I'll finish double-stranded here only. Double-stranded is two types: homologous, non-homologous, and joining. Homologous is using another DNA template, is error-free. Non-homologous is error-prone. Keep those disease there only. Double-strand is ataxia telangiectasia, Blum, and BRCA1, BRCA2. Single-stranded, how do they differ? What do they differ in? Single-stranded, first thing, helicase is everywhere. So we are not doing common like helicase, exonuclease. We are doing uncommon. How is this different from the other? Base is attached to the sugar by glycosidic bond. So that's why glycosylase is unique to base excision repair. And so on, you have to add the unique finding. Base excision is only long patch and short patch. Long patch because long means in a leading strand, you needed epsilon. So in long patch, epsilon kicks in rather than the short patch. Again, beta is unique. So what is unique is important. So when you say long and short patch, you're talking about base excision. These buzzwords should come to your mind. Polymerase beta, base excision. So how many times you can bring in the differences? Much better, right? AP site, base excision repair. Because only when glycosylase removes the base, you create AP site. This is the AP site. We have not talked about disease clinical syndrome in base excision repair. We have talked about clinical syndrome in nucleotide excision repair. This is still single-stranded thymine dimers. Now here you will not see glycosylase. So see what are the different enzymes that kicks in. And I will just tell you the main difference is in the endonuclease usually or polymerase. Endonuclease, polymerase, or other different proteins. Please bring them in. Mostly, it's not like that one protein works for all. What works for all is only helicase and ligase. Have we done two types of nucleotide excision? Yes, xeroderma pigmentosa is not with transcription coupled, but global genomic. Melanoma, xeroderma pigmentosa, single-stranded. Global genomic, more common versus transcriptional coupled. Please know that sunlight or sun sensitivity may be seen in all. See what other features add or point towards that clinical condition. We have done xeroderma pigmentosa, cocaine syndrome, mismatch. Again, what is common in mismatch? DNA polymerase beta is only for base excision, but look at this delta. The delta comes in, this delta comes in everywhere. Usually delta is the most common polymerase. Mismatch, you have done Lynch syndrome, right? Mismatch, how they are different? They are mismatch repair enzymes. When P50, when there is DNA damage, P53 kicks in, stop the cell cycle, then they take over. All the repair enzymes. Mismatch is one of them. Mismatch enzymes are active when there are a lot of repeats. That is why in mismatch repair defect, you see satellites of different sizes on a gel. So keep single-stranded distinct from double-stranded. Don't mismatch their disorders and their proteins, their enzymes. Homologous is always error-free because you use another DNA template for the repair. Ataxia telangiectasia, Blum, and BRCA1, BRCA2 is double-stranded. P53 is an important break, but to be active, to put a break, it has to be phosphorylated. Another protein apart from DNA damage that does this job is ATM protein. If it's lost, there is no P53 function. That's not good. P53 is essential for the cell cycles to stop whenever there is DNA damage. BRCA1, BRCA2, you associated with breast and ovarian. Blum, please know all the Blum, xeroderma pigmentosa, they are actually chromosomal instability syndrome. Why is chromosome unstable in them? Blum, ataxia telangiectasia, or xeroderma pigmentosa, because the respective repair mechanism is defective in them. Don't forget to bring in the protein defective and the presentation. All I'm trying to tell you is find out what is unique to single-stranded versus double-stranded. Double-stranded disease do not mix with single-stranded. Single-stranded, among them, don't mix and match their proteins, which are unique to each one of them. If you're talking about mismatch repair enzyme, don't call it base excision repair. Just be mindful and read them. Right? That's all. If you have any question, I can open it now. Hope this helped. If you want to listen to the question, you can stay. Otherwise, do we not know to the many thanks? Yeah, you're welcome. Do you know this? No. You don't need to know repeats. No, we have not taught you repeats, please. Yes, Esther, tell me.
Hi, so I just had a general question. So what are some important things we need to know from the case studies? And my second question is, you said we don't have to really know all the clinicals for the genetics.
Yeah, for
and so-
inheritance, not genetics, please. Don't take
oh,
me wrong.
inheritance, yes.
Yeah, inheritance pattern, just
Yeah,
to clarify. For CV, just read the CV and the question.
and the
Maybe
questions, yes.
question, yeah.
Okay, perfect. And then, so we don't have to know the symptoms for the inheritance.
No.
Like the quiz
No.
questions.
No.
Okay,
You don't need
perfect.
to, because what happened in old curriculum, there was biochem, then genetics, but now it's flipped. It's first genetics and biochem, which makes sense. So that is why, no, you don't need to know the inheritance pattern presentation. Yes, next. Sure.
Thank you.
No worries. Good luck. Yeah. Yeah, but I was asking which induces negative supercoil. If I'm not wrong, it's topoisomerase 2. I'm not asking positive supercoiling. I think my question was what induces. Only in DNA gyrase. DNA gyrase is prokaryotic topoisomerase 2. Okay. You're welcome. Good luck to everyone. Yes, any question? Yes, Jasmine?
So I'm a little bit confused about the difference between pleiotropy, locus, and allelic heterogeneity. Can you go
Very
over what
good. So you are confused between locus and allelic heterogeneity. Yes, you're most welcome, Karan. Now, the difference between, I'll give you one example. Let's take an example of high phenylalanine levels in a patient. Now, high phenylalanine levels, phenylalanine is an amino acid. It should not be high. We found that phenylalanine levels were high in a patient. So what happened is patient in the genetic people want to determine what is the cause. It's an inherited disorder. It's in a newborn, for example. The phenylalanine levels are high. So to find out, they did the gene study. And they found out that the phenylalanine levels could be high because of the enzyme defect or the cofactor defect. This is known as locus heterogeneity. Locus heterogeneity means the clinical presentation is one. That is high levels of phenylalanine, but it may be because of two gene involved, which is enzyme or cofactor, right? Did you understand locus heterogeneity now? Yes?
Okay.
And now allelic.
And you said the two genes involved were gene and what was the other one?
So high phenylalanine levels is one clinical presentation, but it can be due to two loci or two genes. One which is encoding the enzyme, or the other which is encoding the cofactor. So that means this is locus heterogeneity.
Okay.
Now, what is allelic heterogeneity? Now, just as you know that high phenylalanine levels can be because of the enzyme or cofactor. So heterogeneity means you have to make something stable. What you are saying is genety means that should differ. So when you are saying locus heterogeneity, there should be two loci. When you say clinical heterogeneity, there should be more than one clinical presentation. So now when you say allelic heterogeneity, that means more than one allele in a loci should be involved. So that means you have to make locus constant. So let's take one locus, which is enzyme defect. So that is one loci. An enzyme defect may end into three clinical presentation, and it happens in enzyme defect. So what is differing here? If locus defect was an enzyme and you had three different presentation, what is this type of heterogeneity? If there was
That would be allelic?
No.
Because there's three different presentations?
No. I'm saying there is one loci. So first go back. There's one clinical presentation. So clinical presentation is constant, but more than one loci involved. So what is different here? Locus. Okay, now you take one locus and has three clinical presentations. So what is this type of heterogeneity now? What is different here? Clinical heterogeneity. It will be clinical heterogeneity because there was one loci, one gene, but it represented in three different ways.
So, sorry, can I ask you a question?
Again, so there is one clinical presentation and more than one loci involved. So what was different? Locus. So it's locus heterogeneity, right? Yes? Now go further. One locus and three clinical presentations. What should this be? What is different here?
So will be allelic?
It will be clinical heterogeneity, which is not in your terms, but how these clinical presentation differ from each other is because of different allele involved in the locus. That is allelic heterogeneity. Do you understand now?
So clinical heterogeneity is the same thing as allelic heterogeneity.
No. Clinical heterogeneity means one locus has three different clinical presentations. But when they say how do these three clinical presentations differ is because of different allele involved. When there's that statement, then there is allelic heterogeneity. Heterogeneity means whatever they are saying, they want you to choose should be different. Allelic is a single gene when we did Allelic is within a locus. There may be different allele. Who's asking me this question? There may be different allele within the locus also. That is allelic heterogeneity, Lumen.
So could
Locus
you
is,
give an
yes,
example?
different. Yeah, you're very right. Locus is different genes, and allelic is different allele within the locus, though. Yes, within a single gene, different alleles, yeah? Rumen, that's correct.
So could you give examples of like locus versus allelic heterogeneity?
I just gave you, right? High levels of phenylalanine, more than one
Okay.
locus involved, one locus, 3 clinical presentation, and how do they differ from each other? Allelic heterogeneity. I think all this heterogeneity to choose depends on the statement in the question stem. You just get my point? Whatever they are asking should be different. If they want to choose you to choose allelic, they should mention that allelic is different and locus is common. If they want you to choose locus, clinical presentation should be common, locus should be different. If they want you to choose locus versus allelic. Do you understand my point? Whatever they are asking you to pick should be different in the question stem. Oh, yes, same presentation. BRCA1, BRCA2 are involved, genes involved in the same repair, but with different genes involved in the same repair. It's not a disease, it's a cancer. They both are, the BRCA1, BRCA2 defect leads to both. Yes, they are different genes, but lead to breast and ovarian cancer. It's not a disease, please. Allelic can be different mutation in the, absolutely. So see, allelic means they are different within the same locus or gene, absolutely. Allelic can be different allele within a single gene. Yes, love Kataria?
Hi,
Yes,
I had a question about the manifesting heterozygote.
yes.
Will we see those in pedigrees, or is that something that's just separate?
Yes, you can see manifesting heterozygote in the pedigree. Dr. Hansha has very beautifully explained there. She has explained in one of the PowerPoint slide. You can see manifesting heterozygotes. Anybody has the slide? I can just tell me the number, but she has. Yes, manifesting heterozygotes is for X-linked disorder. And Dr. Henshaw has very beautifully explained you can infer manifesting heterozygote from a pedigree, absolutely. Anyone has the number, I can show you, but I don't know number off my head now. I think it's slide 10 where X-linked recessive. You need to, one thing I would tell you is you have to read your lecture slides, please. Your lecture slides and PowerPoint are heart of your questions. I'm just giving you a focus, a chart, right? Let me pull that slide. Is it X-linked recessive and slide 10? Let me see the chats, right? Is it as X-linked recessive? Okay, I will clarify. Let me do the sex-linked inheritance and slide 10. Can I do that? Then I'll come to you, Kevin. Give me a minute, please. Yes, P53 is a tumor suppressor gene. Let me pull out the slide, please. Please read your lecture slides, please. Believe me, there's no shortcut to success, right? Yes? I can give you an overview, but more details you master, better grades, believe me, better grades. So let me pull out that slide and explain you how you can see pedigree from a, how can you do manifesting heterozygote from a pedigree. Let me see if I can pull it out quickly. X-linked recessive. Give me a minute, please. Sorry, I was not prepared for this. But I'll tell you, since you have a question. I hope it's slide 10. Let's see if I'm sharing the slide. Let me pull out slide 10. Everybody, but is it slide 10? I don't think so. I think so, yeah. Let's do this. You're with me? Give me a minute. Okay, let's put slide 10. Let's see if we can, which is this one, okay? Now, let's look at this. Okay. I think it was not in this. She has a question. She has a question where and I don't think so. This is slide 10. Slide 10 is just showing what is manifesting heterozygote. I think she has a question. Let me see if I can see that. Where by pedigree, you can see that it is manifesting heterozygote. Anyone has any idea? It's in a question stem.